I will stress at certain points that is very important for NEET examination. That is chapter 13 in first PSC, photosynthesis in higher plants. So what is the definition of photosynthesis means? So green plants in the presence of sunlight, they prepare their own food by combining the carbon dioxide and water. That is 6CO2 plus 12H2O gives C6H12O6 plus 6H2O and 6O2. Oxygen is released during this process. So photosynthesis can also be defined as it is a dye-sensitized photochemical redox process. Dye-sensitized means chlorophyll sensitized. Dye-sensitized. Photochemical, light energy is converted into chemical energy. Photochemical redox process where the reduction and oxidation takes place. Reduction of carbon dioxide takes place and oxidation of water takes place during photosynthesis. This is very, very important. And the oxygen released during photosynthesis comes from water alone, not from carbon dioxide. Here 12H2O, 6O2. Only water, not from carbon dioxide. This is also important for the NEET examination. So coming to the chapter begins with what are all the factors required for the photosynthesis. So chlorophyll is essential for photosynthesis. So to this, take a variegated leaf in a plant. Select a variegated leaf. That is in a croton plant where you can see the different patches of green, yellow and all those things. Cover one leaf and another leaf, expose it to the sunlight in a potted plant. So the covered portion does not show the presence of starch. Only the green parts here shows the presence of starch. Starch with iodine gives blue color. This shows that chlorophyll is necessary for photosynthesis. Another experiment given in the textbook is the next experiment. Van Mole's half-leaf experiment. This experiment shows the carbon dioxide is necessary for photosynthesis. Take a well-watered potted plant having the long leaves and insert half of the leaf into a bottle containing KOH and place the pot and the apparatus, the bottle containing KOH in the sunlight. After some time, take out the leaf and test for the presence of starch. Half of the leaf present inside the bottle doesn't show the presence of starch. This is because KOH absorbs carbon dioxide and made it unavailable to half of the leaf. But half of the leaf present outside the bottle shows the presence of starch. This shows that carbon dioxide is necessary for photosynthesis. Many experiments began in the year 1770 itself to show that atmospheric air plays an important role in photosynthesis and even in growth. But in 1774, Joseph Priestley discovered oxygen. Ever since he discovered oxygen, he started experiments to show that how the oxygen is released from during photosynthesis from the green plant. Rat pot experiment. He took a potted plant and he placed the burning candle near it and enclosed a bell jar over it and inserted a rat. Rat is allowed to enter into this kind of bell jar. So where the potted plant is kept, there, the rat stayed alive and the candle burns. But he repeated the same experiment without a potted plant, just a rat and the candle. The rat after some time gets suffocated and the candle extinguished. This shows that oxygen is released during photosynthesis. An elegant experiment did by the engine house to show that light is necessary for photosynthesis. He took a plant, aquatic plant, and then when exposed to light, more bubbles. He observed the bubbles coming out. This shows that oxygen is released during photosynthesis. Here the plant placed in water where in the dark place no bubbles appear. This shows that light is necessary for photosynthesis. Then it was in the year 1854 that Julius Sachs showed that the glucose is required for all the activities, and food in the plant stored in the form of starch. That's the contribution of Julius Sachs. And T.W. Engelmann, he showed that two kinds of light necessary for photosynthesis. So he did an experiment, took an alga that is called Cladophora and placed Cladophora in a suspension containing aerobic bacteria, and then he exposed this the suspension containing plant to different kinds of light. Only in the presence of red and blue lights, the maximum bubbles comes out from the plants, and the aerobic bacteria collects at the where bubbles coming out. This shows that two kinds of lights very essential for photosynthesis, that is the red light and blue light. And next to this, it was Cornelius van Niel. He did an experiment to show the bacterial photosynthesis. So in bacteria like green, purple and green sulfur bacteria, and then the raw materials required for the bacterial photosynthesis are the carbon dioxide and H2S. It is not a water which is a proton donor. Here the proton donor in bacterial photosynthesis is H2S. And oxygen is not released during this photosynthesis. This is very important for your NEET examination. Raw materials required are the carbon dioxide and H2S, and the products formed are CH2O, that is carbohydrate, water and sulfur. That experiment did in purple and green sulfur bacteria. The equation is CO2 plus 2H2S. CO2 plus 2H2S gives CH2O, and then H2O plus 2S are the products. So oxygen is not released during the bacterial photosynthesis. Thus, the bacterial photosynthesis consists of only PS1, not the PS2 at all. And then the various factors which are required for photosynthesis. Light. All the light reaching to the earth is not used for photosynthesis. Only a 2% of it, that is PAR, photosynthetically active radiation, PAR 2 to 10, is useful for its photosynthesis. Light of all the wavelengths is not used for photosynthesis. And if the wavelength is more, the amount of heat liberated will be less. For example, in the presence of infrared rays, photosynthesis will not take place. If the wavelength is less, the heat liberated will be more. In the presence of X-rays, UV rays, photosynthesis will not take place. For the photosynthesis, the wavelength should be in between 400 nanometers to 700 nanometers. That is VIBGYOR. In the presence of the VIBGYOR, the photosynthesis takes. But the maximum photosynthesis takes place in the presence of both lights, red and blue light. If alone in the presence of red light, but both in the presence of red and blue light, the maximum photosynthesis takes place. This is very, very important. And the saturation for the light is the 10% in plants. That is also very important. And coming to the chlorophyll pigments, also the factors required for the photosynthesis. As I told you earlier, plastids are present in the plant cells, and these plastids which are colored are called chromoplasts, green colored plastids are chloroplast, colorless leucoplast. And the chloroplast, the green colored plastid, consists of two layers, inner layer and outer layer, and green colored grana and colorless stroma, and there is the stroma lamellae which connects the grana composed of thylakoids, plate-like structures called thylakoids, which are membranous thylakoids. And in the stroma, double-stranded DNA, 7 step ribosome and certain enzymes are present, in the chloroplast, and then this grana, it consists of both chlorophyll, different pigments like A, B, C, D, etc., and accessory pigments like the carotenoids. Carotenoids consist of xanthophyll and carotene together, and they absorb sunlight, pass on to chlorophyll A. Chlorophyll A is the reaction center that is in photosynthesis, and only the chlorophyll A has the ability to convert the light energy into the food energy or the chemical energy. Chlorophyll A has blue-green color and chlorophyll B has yellow-green in color. So far not asked. This is also important. And the carotenoid xanthophyll yellow color, yellow to orange. Xanthophyll is yellow in color, and the carotene that is yellow to orange in color. Together called carotenoids, xanthophyll and carotene are the accessory pigments. They protect the chlorophyll A from heavy light damage. And the other factors which are required for photosynthesis are the electron acceptors like the pheophytin, plastoquinone, plastocyanin, ferredoxin reducing substance, ferredoxin, and the cytochrome B, F are the various other factors required for photosynthesis. And the raw materials required for photosynthesis and the carbon dioxide and water. So carbon dioxide and water are the raw material required for photosynthesis. Water acts as a proton donor, and the carbon dioxide gets reduced. The saturation point of carbon dioxide is 360 microliter lubricase inverse. 360, this is important, microliter lubricase inverse for C4 plants, and it is 450 microliter lubricase inverse, that is in the C3 plants, that is the saturation point of the carbon dioxide. But there are certain crops which grow very well even if the concentration of carbon dioxide is small. They are called greenhouse crops. Greenhouse crops, that is bell pepper and tomato. Bell pepper and tomato. This is also very important for the NEET examination. These are the various factors which are required for the photosynthesis, coenzymes, even coenzymes. They function catalytically, be reduced or oxidized alternately, that is NADP. So the assimilatory powers are ATP and NADPH2. And this photosynthesis takes place in two steps, light reaction and direct reaction. Light reaction requires light, and the end products of light reaction are the ATP, NADPH2, water and oxygen. Water and oxygen are the end products of light reaction. And the light reaction, dark reaction takes place in the stroma of the chloroplast, and the end product is the glucose. So light reaction, it involves photosystem 1 and photosystem 2. Photosystem 1 is cyclic photophosphorylation. So in a cyclic photophosphorylation, electron ejected from the chlorophyll returns back to the chlorophyll. In PS2, non-cyclic photophosphorylation, virginal electrons ejected from the chlorophyll do not return to the chlorophyll. They are used for the reduction of NADP into NADPH2. That is non-cyclic photophosphorylation. So PS1 is shorter than the PS2, but PS1 takes place both in grana and stroma lamellae. This stroma lamellae does not contain NADP reductase. That is very important for your NEET examination, stroma lamellae. Because in stroma lamellae only PS1 takes place, and in grana both PS1 and PS2 takes place. PS1 and PS, that's grana consists of NADP reductase, but in stroma lamellae no PS2, hence no NADP reductase, and NADP reduction takes place only during the non-cyclic photophosphorylation. So in PS1, a light having the wavelength of 700 nms involved and 680 nms, and no photoionization of water. Photoionization of water takes place, not connected to OEC, oxygen evolving complex, connected to OEC, oxygen evolving process. The end products are only ATP in the PS1, and the end products are the NADPH2, water, oxygen, and even electrons for electrons. That is the products of this non-cyclic photophosphorylation. This can be summarized as following. Both the photosystem 2 and photosystem 1 are combined in higher plants. In a photon of light, to start with, to begin with the PS2, in a photon of light having the wavelength of 680 nms hits chlorophyll molecule gets excited, releases electrons which reach pheophytin or electron acceptor, not given the pheophytin name, so electron acceptor. Then the downhill process, that is plastoquinone and cytochrome B and cytochrome F, plastocyanin, and enter into PS1 and reach FRS and FD, and NADP gets reduced. When the photon of light 680 nms hits chlorophyll molecule, gets excited, releases electrons, as I told you earlier, electrons goes to pheophytin. But during the electron jump, energy is released. This energy released during this LHPC complex is formed because of the energy release. That is light harvesting protein chlorophyll complex. This splits water into H plus and OH minus ions. For example, 4 molecules of water is taken, then it gives 4 H plus and 4 OH minus. This kind of splitting up of water called photoionization of water observed by Van Neel. And here the 4 H plus takes the pathway of non-cyclic photophosphorylation and released into the lumen of thylakoids. That is very, very important. Lumen of the thylakoids, H plus ions, protons are released, and the OH minus along with OEC, oxygen evolving complex, that is ZMN complex, not zinc, just ZMN, along with OEC releases oxygen. So 4OH minus, it gives 2 molecules of water, a molecule of oxygen, and 4 electrons, and these electrons return back to the chlorophyll through chlorine. That is, we discussed in the previous chapter, that is the mineral nutrition, that is very important. But thus oxygen is released during photosynthesis, and it takes place during the non-cyclic photophosphorylation, and all the oxygen released during photosynthesis comes from only water, not from carbon dioxide. Again, this is very important. And in the lumen, ATP synthase enzyme is present, or ATPase enzyme is present. This enzyme consists of 2 parts, F0, a channel, and F1 present outside, that is in the stroma, and through which the H plus ions move out, and resulting in the formation of ATP takes place, and H plus ions in the presence of the NADP reductase. It gives rise to NADPH. NAD gives rise to NADPH2. And here that is about how the H plus ions move. And as I told you earlier, the virginal electrons ejected from the chlorophyll goes to the electron acceptor pheophytin, plastoquinone, cytochrome B, F, and this is uphill process, downhill process, and again this gives Z scheme, that is very important. And then ferredoxin reducing substance which contain iron and sulfur, as I told you earlier in the previous chapter, and then so ferredoxin, NADP plus these electrons, say for example 2E, 2E, and then R 4E, and then this 4H plus plus 4E minus here, E minus gives 2 NADP, gives 2 NADPH2. Thus the virginal electrons ejected from the chlorophyll do not return to the chlorophyll. They are used up for the reduction of the NADP to NADPH2 assimilated power. And in the cyclic photophosphorylation, when a photon of light having the 700 nm chlorophyll molecule gets excited, releases electrons, goes to FRS, ferredoxin reducing substance, and FD, ferredoxin, and ferredoxin to cytochrome B, ATP formation takes place because of chemosynthetic that is the chemoosmotic synthesis. So chemoosmotic synthesis, that is ATP formation, and then again from cytochrome B to cytochrome F, again ATP formation takes place. ATP formation takes place, and chemoosmotic hypothesis proposed by Mitchell, that is during as the electrons move from cytochrome B to F, energy is released. This energy is used for the formation of ATP that takes place in the presence of oxygen, hence called photophosphorylation. That is about the chemoosmotic hypothesis. Thus, only the ATP are the products of the PS1, whereas the NADPH2, water, oxygen, 4 electrons are the products of non-cyclic photophosphorylation. The products of non-cyclic photophosphorylation are used up during the direct reaction. This direct reaction takes place only in the stroma of the chloroplast. RUMP, ribulose monophosphate, present in the stroma, and the end products of light reaction used up during the direct reaction, that is biosynthetic phase. This direct reaction is also called biosynthetic phase. So this RUMP, about 6 molecules of RUMP, 5 carbon compound present in the stroma. By using the 6 ATP molecules, so that is the products of light reaction, gives rise to 6 molecules of RUBP, that is ribulose biphosphate, where 6 molecules of ATP used up, and 6 molecules of RUDP or RUBP in the presence of an enzyme RUBP carboxylase gives unstable compound or 12 molecules of PGA, phosphoglycerate, the first stable carbon compound produced during the direct reaction. As I told you earlier, in the nitrogen cycle, the first stable compound is ammonia, and here the first stable is 12 molecules of 3 carbon compounds, phosphoglyceric acids, and phosphoglyceric acids are phosphoglycerates of the 12 molecules, and then these 12 molecules of phosphoglyceric acid gives rise to 12 molecules of PGAD by utilizing 12 molecules of ATP and 12 molecules of NADPH2. Thus, for the reduction of 6 molecules of carbon dioxide, 18 molecules of ATP and the 12 molecules of NADPH2, very important, are required. Then it gives rise to PGA, gives rise to PGALD, phosphoglyceraldehyde. 12 molecules of the 12 molecules of PGALD, 2 molecules gives rise to a molecule of glucose, the end product of the dark reaction, and 10 molecules of PGALD, 3 carbon compounds gives 6 molecules of RUMP, and cycle will be repeated. Thus, the dark reaction takes place in the stroma of the chloroplast, and Melvin Kelvin discovered this dark reaction. It takes place in 3 steps: the carboxylation, reduction, and regeneration. Carboxylation, reduction, and regeneration, and these things also very important. Very important. And then here the first stable carbon compound in majority of the plants, in C3 plants, are the C3 compound phosphoglyceric acid, PGA. And then whereas in few plants, in majority of the monocots, especially in the Zymers, the first stable carbon compound produced C4 compound, oxaloacetic acid, maybe malic acid or aspartic acid. This pathway is called C4 pathway. This takes place in the presence of an enzyme PEP carboxylase. In the presence of PEP carboxylase, phosphoenol pyruvate, the 3 carbon compound, by fixing the carbon dioxide in the presence of PEP carboxylase, gives 4 carbon compound OAA, and then or malic acid or aspartic acid, and they enter into the mesophyll. From the mesophyll they enter into the bundle shaped mesophyll. There in the bundle shaped mesophyll they release carbon dioxide and return back to the mesophyll. Thus the C4 pathway takes place in mesophyll, and this carbon dioxide released again used up by the plants used up by the RDP carboxylase to synthesize the glucose. Thus, the carbon dioxide will be fixed twice, and these C4 plants, they have peculiar anatomy called Kranz anatomy, wherein the bundle sheath, the border parenchyma is called bundle sheath mesophyll, as it contains a granule chloroplast, a special type of chloroplast. And the C4 pathway takes place in mesophyll, that is very important, and C3 pathway takes place in bundle sheath mesophyll, though they have a different structural arrangement. So this is very important as far as the C4 pathway is concerned. And there is no photo respiration in C4 plants, because the concentration of carbon dioxide will be always more in C4 plants, and if the concentration of carbon dioxide will be less, then photorespiration takes place. Three cell organelles play an important role in photorespiration, that is the peroxisomes, mitochondria, and chloroplast. And as the ATP and NADPH do not synthesized during this photorespiration, it is a wasteful process. So this takes place in the presence of an enzyme, RDP rubisco, that is ribulose biphosphate carboxylase oxygenase. The rubisco, when concentration of carbon dioxide is more, it gives rise to PGA. If the concentration of carbon dioxide is less, it gives one molecule of PGA and one molecule of phosphoglycolate, which is 2-carbon compound, that is the product that is formed during this photorespiration. Anyhow, it is the phosphoglycolate, the 2-carbon compound, and it is a wasteful process. Takes place only in C3 plants but not in C4 plants, because of concentration of carbon dioxide is more. So with this in detail, I completed the chapter number 13, photosynthesis in higher plants, very important for your examination. Please subscribe.